Ask a biologist how cells turn DNA into protein, and I bet they'll say to you, "Oh, yeah, we know how that works. Imagine you were a cell, and I'm your DNA. I'm going to hand you a 10-page instruction manual on how to build some protein. But before I give it to you, I'm going to rip out 9 of the pages and say, oh, you actually need that one." That's how human genes work. They synthesize a whole big long message and then throw away most of it before it actually gets to the protein factory, the ribosome. So, now knowing that, imagine I am the cell and you are a molecular biologist. Say you want me to make some ton of protein, either to study it or to use it as a drug. What are you going to do? You are going to give me a ton of DNA, right? But hey, you are a scientist, you are clever, you're not going to waste your time making the whole big long message when most of it's just going to get thrown away, right? You could just give me a little pieces that go to the ribosome called the exons, right? Well, in that case, I, the cell, would turn around and say, here's your protein, smartass, and just give you a little bit or sometimes none at all. What's up with that? Well, you should have given me the whole gene because those bits, they get cut out. They're called introns. And it turns out that even though they literally gets destroyed seconds after they're built in the cell, most genes need at least one intron for effective protein production. How's this possible? Well, that's what I'm going to try and figure out. Does the intron sequence matter, or is it just ripping up something that's important for the cell? Well, now picture two cells. You give them the same gene, same exons, two different introns. Cell 1 gets intron A, cell 2 gets intron B. Who will make more protein? Well, people have done this experiment, and you know what they saw? Sometimes it doesn't really matter. Sometimes one intron will give you 500 times more protein than the other. So here's my plan. Instead of two cells, I'm going to take a million of cells and million of different introns, each with a unique randomly generated sequence. Then I'm going to measure and see who makes the more protein and what intron sequences differentiate the strong introns from the weak one. Because I want to learn a few words of their language and then use to ask them what mysterious chemistry allows them to speed up a production in a factory that I will never touch. Because I think the problem is we already speak the language of exons, the genetic code, how DNA goes to protein, whatever. We think that's the whole story, but we have no idea what those introns might be whispering in the ear of ribosome long after they are gone. And I think it's about time we started to listen, because I bet they have a lot to say. Thank you.